Okay. I'm gonna work through this and see if this works. Okay, so should we give it a minute, maybe?
Share another microphone.
I don't think it's the microphone.
Oh, Drew.
Easy, I'll hit the beach with your fingers.
That's Drew trying to call in.
That's what Gary.
Working.
Yeah. He's got a 1-year-old, so.
Okay, well, with that background music, so I'm just going to talk a little bit about the role of ILCs in allergic inflammation. Like I said, we've kind of had a pretty good discussion about that as well. But just to kind of review, allergic responses are characterized by the production of type 2 cytokines by the Th2 helper cells. Can lead to eosinophil and mast cell degranulation, goblet cell hyperplasia, mucus production, increased IgE levels, as well as smooth muscle contractions. And these can be developed erroneously to kind of repeated low dose exposure to innocuous allergens, causing disease states. And those are just some examples of the allergens we deal with. And again, the Th2 cytokine responses are what's driving the allergic diseases, including asthma, allergic rhinitis, atopic dermatitis, as well as food allergy. The IL-2, IL-5, IL-9, and IL-13 are the important cytokine players. And let's see, what's the last point?
When did 9 become important?
What's that?
IL-9, not in my list of important ones.
Oh,
It was must including must.
oh, okay, great. Yes, I am not sure what IL-9 is for. Okay, and then what is this? Okay, so the ILC2s respond to the IL-23, the IL-33 that we were just talking about that play a really important role, as well as the TSLP, another factor at both the mucosal and the skin surfaces, and then they cause the Th2 cells to secrete all these cytokines and cause these reactions at the different barrier surfaces that we talked about. And this is another pretty diagram that shows that. Or actually, this is a small part of the old diagram that I showed. And so in corollary, the ILC2s also, because of their similar cytokine profiles to the Th2 cells, cause a lot of the same effects. And this is just to kind of highlight the other roles that they play, like I mentioned earlier, the helminth infection, as well as influenza. It essentially helps to kind of heal the lungs after influenza viral induced lung injury. And influenza specifically, because that's like one of the studies they've done, tested it on, I'm sure it will, it plays a role in different viral induced lung injury as well. All right. This is a diagram that kind of shows how the ILC2s interact with the T and B cells and the barrier surface cells. So with exposure to helminth or allergens, these cells, the innate immune system comes in, does this thing, and then releases a whole bunch of regulating factors, including IL-33, which is important, and then these are the other two that I kind of mentioned. And they, in turn, affect the ILC2s, as well as, obviously, the adaptive responses. And they produce these effector molecules that go on to affect the effector cells and leads to the Th2 cytokine-mediated inflammation. As well as also kind of stimulating the Th2 cells they correspond with as well. Okay. Yeah, I mentioned this before. This is a kind of diagram that shows the role of ILC2s in the human allergic diseases, both in human models as well as marine models. And these are just some of the ones that we are particularly concerned about. So I thought I would highlight them. And then over the next several slides, I will kind of briefly highlight some of the studies that have been done in investigating the roles of ILCs in these specific diseases. So ILC2 and asthma. So in humans, elevated levels of IL-25 and IL-33 have been shown in a human asthmatic lung tissue. You can also see peripheral blood ILC2s that respond to asthma-associated prostaglandin levels by producing IL-13 that cause downstream effector inflammatory effects. And some lung ILC2s have been shown to express a receptor, leukotriene receptor 4, and ligation of this receptor can induce IL-5 production by the ILC2s, and that stimulates the eosinophils and causes the downstream inflammatory effects. So montelukast, which is a leukotriene receptor antagonist, can interfere with this process and might be part of the reason it's effective in asthma control, as well as its other effects. So essentially, this could be a relevant target in the treatment of asthma in patients. Okay, so this is a paper that is currently in press in the Journal of Allergy and Clinical Immunology. I'm not going to go, very in depth in the specific methods and research that this team does. I just kind of wanted to highlight some of the different researches in different allergic diseases that's going on. So essentially in this study, they had two types of knockout mice. One was deficient in T and B cells but had normal ILC numbers. And then the other was deficient in T and B cells as well as ILC 2 specifically. And they transferred the bone marrow from these knockout mice to irradiated mice with chronic asthma. And they were able to show that, so this is the T and B cell deficient only cell, and this is the T and B cell and ILC 2 deficient cells. So they saw that the mice that received the marrow from just the T and B cell knockout mice, so which is the RAG1, had maintained kind of airway hyperreactivity, but that mice that received marrow from the knockout mice that was also deficient in ILCs lost the airway reactivity here.
Do you remember about how long after transplant that was? Getting at what is the half-life of these cells in the tissue? Are they short-lived, long-lived?
Like how long the transplant was after they were irradiated?
No, I mean after they transplant them, how long till they do the methacholine challenge?
I'm not sure. I remember.
Or do you generally know what the lifespan of these cells is in the periphery? Because I mean, this would obviously imply that they had to wait until all of the
reconstituted. Yeah. I'm not sure.
So these were mice that already were an asthma model. They had asthma. Mm-hmm. And the point is, if you give them cells that lack T and B and innate immune cells, they repopulate their lungs with healthy, non-inflammatory cells, and their asthma goes away.
I don't know about the repopulating with healthy lungs, but they don't have the active inflammation causing the hyperreactivity that the cells that still have the innate lymphoid cells do.
Well, I guess they're implying that, just having the ILC2 is sufficient.
Yes. To still cause hyperactivity.
Perpetuate the inflammation.
Yes.
Actual cells.
Yes.
So everything we've thought before, that Th2 cells are enough to have asthma, appears to be wrong from this model. You need Th2 cells and ILCs.
Well, they didn't do the Th2, they didn't do Th2 like positive and then ILC negative. So essentially, ILC alone can cause asthma.
Do you remember finally? IL-9 is also signaled through the common gamma chain. I think it's also there, so they won't have any mast cells in their airways as well.
What's the point, David?
That they won't have any mast cells in their airways, if that's right. So if they won't have mast cells, maybe it's not the innate lymphoid cells, it could be other cells.
It's just entirely too complex.
Let's move on.
Also fairway responsiveness.
For the initial response.
Yeah, because they've done mast cell knockouts and you get the same airway responsiveness.
You don't need them.
Was it this hard when we were fellows, Gary?
Much too complicated. It's time to retire.
I can evaluate my key right now.
So then, looking at ILC2s and its role in chronic rhinosinusitis, this is actually a little bit interesting to note that apparently chronic rhinosinusitis patients with nasal polyps, their kind of inflammatory process is different from chronic rhinosinusitis patients without nasal polyps. So without nasal polyps, kind of operate through a Th1 cytokine profile, and then with the nasal polyps, they are through a like Th2 cytokine profile. So in this study in the American Journal of Critical Care Medicine that came out in August 2013, researchers essentially looked at the role of IL-33 and what they call IL-33 responsive innate lymphoid cells, which are the IL-2s, because this preceded the nomenclature, I think, and looking at their role in chronic rhinosinusitis. So they use quantitative PCRs to measure the expression of one of the chains of the IL-33 receptors, which was noted to be elevated in the chronic rhinosinusitis patients with nasal polyps. And they use flow cytometry to measure the ILC2s and also show that there was an increased percentage of innate lymphoid cells, like ILC2s, in CRS with nasal polyps as compared to the patients without nasal polyps. And they don't really know why one is driven with the Th1 kind of profile and the others with the Th2 profile. But it was something that they found different. I thought that was kind of interesting.
That makes me wonder, do we know how these cells respond to pharmacologic agents like cyclosporin and other cells that we traditionally think suppress T cell function? Do we know anything about the pharmacotherapy?
I did not see any studies on pharmacotherapy of these targeting these cells. And then their role in atopic dermatitis. Okay, so this is a paper where I just really kind of included for the pictures. They looked at the IL-25 and its role in atopic dermatitis and skin barrier functions. So again, atopic dermatitis operates kind of through a Th2 response and has increased levels of Th2-associated cytokines and IgE. This study came out in 2011, and researchers looked at skin biopsies from involved skin and uninvolved skin of patients, comparing like the same patients, of people with atopic dermatitis against the control. And they saw that they stained for the presence of IL-25, which is one of the regulating factors of ILC2 that we saw earlier in all the diagrams. And they note that I don't know if you can see very clearly that there is increased staining of the IL-25 in the involved skin of patients with atopic dermatitis versus the uninvolved as well as the control. IL-25 also acts to inhibit kind of filaggrin synthesis, which affects the skin barrier function in atopic dermatitis as well. So it kind of plays like it activates the ILC2, and then it also affects the skin barrier in patients with atopic dermatitis. And this is in patients, staining in patients with contact dermatitis, the involved skin and the uninvolved skin. Okay, so in summary, while we're learning more and more about the roles that ILC plays in the physiology and the pathophysiology, I think we're really just kind of touching the surface. And like David said, it's really hard to kind of see if they're distinct entities or if they are, if there are more of them, and how to group them in the future besides just from their cytokine profiles. One good thing is that they might be new targets for therapy, which is always good for the different diseases that they are involved in. Essentially, there's still a lot to learn for them. Any other questions?
If you go back to the skin cells and you showed the uninvolved skin and involved skin and then a control,
Yeah.
what exactly where is the staining for IL-25? Is it just in that little block?
Oh no, it's the red parts.
Either I'm colorblind.
Yeah, I don't see any red.
Yeah.
Can you see?
So it looks like the uninvolved skin and the involved skin are similar, except for there's more inflammatory cells at the surface of the involved skin. But then the control doesn't have that second band in it like the other two do. So I'm trying to figure out.
So I'm looking at the maybe it's the coloring, but you can see like darker staining in the involved skin, like
So
here.
is IL-25 just being used as a marker for ILC cells? That they seem
Yes.
positive?
Yes, because they cause the development of ILC2s. So yeah, like all here. Can you see it better in here? Maybe the red parts. So in contact dermatitis, yeah. Yes, Dan?
In your reading, has anybody looked at what leads to the increased number of those cells in those specific areas? Is it more as home to it, or are they dividing there? What is it? And it's interesting we don't ever really pick it up in the periphery. I imagine at a CBC you see a blast-like cell circulating around. I think somebody would notice.
Yeah, I think I read that in their development, they kind of migrate to they're found in increased numbers in epithelial cells as opposed to other tissues of the body. So I think they are geared towards specific locations.
That's a good point. I mean, they're originally a bone marrow-derived cell.
Mm-hmm.
They have to get to the tissues. They have to circulate at some point.
Yes.
They don't circulate
They
in
don't
adults
circulate
that we can measure.
Are there lymphomas or malignancies with these types of cells described yet?
I haven't seen those, no.
This reminds me of Seinfeld, where George Costanza's mother is at dinner and they offer her a glass of Merlot. And she says, "Merlot, I haven't heard of it. Is this something new?" At least something new. I hadn't heard about it.
They just know Manischewitz.
Okay.
I also just kind of like to take a minute of your time and tell you guys about something exciting that's happening this week. The largest annual human migration is going to occur this week, and it's for the Lunar New Year, Chinese New Year. So every year, this is the most important holiday in kind of Chinese tradition, a lot of Asian tradition. Everybody has to go home to wherever their family is for especially Chinese New Year's Eve dinner. And because of the population of China is 1.4 billion, you have everybody scrambling to get home this week.
Amazing.
So there are just going to be estimated 2.8 billion journeys over kind of 40-day period, 295 million rail trips, and 57 million plane rides during the holiday. And people have often, because, if they can't afford the plane, they can't get the tickets, or they live in very remote areas, they have to take 5-6 different train rides to get home. But it's really important, and because a lot of people coming from rural areas live in the cities, they all have to kind of migrate back at the same time. And it's going to be the year of the goat. Chinese New Year's is on Thursday. And then thank you to Dan for taking my call for the next 2 days so that I will be able to go home for New Year's just before I head to Houston. All right, thank you.
More than a year.
No, I'm standing both.
And manning the fort here.
Oh, that's clear. Application of a lot of those slides is upstream of that or all these other lymphocytes or something. It's more of an amplified effect. It seems like. Yeah. Part of it,
Yeah. So the approach is not back.
Yeah.
But it's solid. You know, not terrible.
It almost but then to be itself there, you know, whatever, 3 times.
Still getting the soul layer.
I don't. I'm going to be okay. I'm on call.
And you have to sleep on the back. Alright. Hopefully we'll have better
I had just saw you.